Okay, well, let me start by thanking the organizers for inviting me to speak here. It's a great honor. So today I'm going to tell you a bit about microbiome research. And I want to give you an idea about what microbiome data look like and what I think are some of the main statistical challenges in this area. Okay, so let's start by just talking about what the human microbiome is. So the human microbiome is just simply the collection of bacteria that colonize our bodies. So what you're seeing in this very pretty picture is a phylogenetic tree, okay, in a radial representation. And the leaves of this tree are different bacterial species that have been identified in our bodies. And then along the circumference, you can see which tissues in the body they're present in. So, you know, in blue you have the stool, you have, I think, it's a bit hard to read, but, you know, you have cheek and dental plaque, and the tongue, the nose, the vagina, and in the skin. And so there's this huge variety of bacteria that are in our body, and most of them are what we call commensal bacteria. They're not harmful, okay? They just live in symbiosis with us. And so we want to understand what these bacteria do. And we're mainly interested in the most important microbiome seems to be the microbiome in the gut. Okay? And the way that we understand that is through samples of stool. And why is the gut microbiome so important? Well, we have this great diversity of microbes in the gut, which are constantly turning over material. So they're processing, they're metabolizing not only the food that we consume, but also drugs and toxins, everything that goes in the body. And they're they're able to ferment certain food products, special types of carbohydrates that we can't metabolize ourselves. And so they're producing, you know, vitamins and bioamines, a number of important metabolites. And through these metabolites, they interact with the tissues in the gut. So they interact with the epithelial layer of the gut, and which, you know, it signals to produce mucus or to proliferate at different points in time. And it also has a very important interaction with the immune system. Okay, so not only do these commensal bacteria compete with potentially harmful microbes, but they also signal the immune system to upregulate or downregulate the production of regulatory T cells, and that keeps inflammatory processes in check. Okay, and that's considered to be very important. So what happens when the microbiome goes awry, when there's an imbalance from the natural state of the microbiome? Well, you get a number of diseases that are associated to microbiome imbalances. One of the most dramatic examples is infection with a bacteria called Clostridium difficile. And this is a bacterium that causes a chronic infection in patients, and it's very difficult to treat with antibiotics, because when you give antibiotics to a person, that kills a lot of the commensal bacteria in the gut as well. And then when the antibiotic effect wears out, it's very easy for this pathogen to recolonize the gut. And so you have these recurrent infections, and some people used to get very, very sick from it. But it turns out that a fecal matter transplant is an extremely effective therapy for this infection. So if you treat a patient with an antibiotic and then you transplant some fecal material from a healthy individual, that can cure the infection from C. difficile. So there's a very clear relationship between an imbalance in the microbiome and this infection. But the microbiome is also implicated in a number of autoimmune disorders. So the most important are probably the ones that fall in the spectrum of irritable bowel disease. And this is a condition that affects an increasing number of people. And we're just starting to, it's been clear for a long time that there are significant differences in the microbiome of people with the disease and people who are healthy. But we're just starting to understand whether this relationship is a causal relationship, okay, and how we might be able to treat irritable bowel disease by affecting the microbiome. There are a number of other autoimmune disorders like diabetes and rheumatoid arthritis that have been implicated in where the microbiome has been implicated. There are some very interesting links with obesity and metabolic syndrome, especially in animal studies. Okay, so in mice, you can show that if you transplant into a mouse the microbiome of an obese person, they acquire symptoms of obesity. So that's very intriguing data. There's also a connection with cancer. Okay, so for a long time we've known of viruses and bacteria that are associated to cancers, like H. pylori is one that's very famous. It's associated to cancers in the stomach and in the esophagus. And there's also, the virus, the human papilloma virus, which causes cervical cancer. But recently, it's been observed that microbiome imbalances are important. They affect the way that cancer immunotherapies work. Okay, so there's a novel modality for treating cancer, which involves using the immune system of the patient, and the microbiome seems very important in regulating that. There's an increasing list of diseases that have some connection to the microbiome. I put a question mark here because it's not clear whether these relationships are causal or they're just associations, right? So this is still a shaky area, but it's intriguing enough that it's generated a huge interest in the topic. And if you look at, this is the result of a semi-rigorous systematic review of the literature, and if you look at the number of citations in high-quality journals for microbiome-related terms, they've gone up dramatically in the last 10 years. Okay, so it's an emerging area in biology. So what made possible this microbiome revolution? Well, it's the development of very cheap sequencing, DNA sequencing technologies. So I say that this is a culture shift, and this is a bit of a pun, because it represents a shift from an understanding of of microbes in the body that was based on taking samples from the body and then culturing harmful bacteria, okay, in a lab setting, and then characterizing those bacteria, to a paradigm where we're characterizing the entire DNA content of the microbial community and seeing which species are present in the sample. So the way that this was done initially, at least, is through this type of pipeline where you take a sample of bacteria, you extract the DNA of the bacteria, and then you amplify a gene called that codes for the 16S ribosome. And this is a gene that's highly conserved in different bacterial species. So every bacterial species will have this gene, but it's slightly different in different species, and so it serves as a sort of marker for a bacterial taxon. And so you can extract the DNA, you amplify the specific gene, and then you run this mixture of genes through a sequencing machine, and that allows you to count how much of each species is present. Okay, so this type of experiment tells you about which species or taxa are present in the sample. Okay, I say taxa because you may be talking about, you may be classifying bacteria at a different level of the phylogenetic tree. Okay. More recently, a new experimental paradigm has emerged where we bypass this amplification step and we simply sequence the entire genome of the mixture of bacteria. Okay, this is called a metagenome because it's not a single genome for a single bacterial species, but it's some sort of mixture. And so in these experiments, you're sequencing the entire genome. You're getting information not only about which taxa are present or which species are present, but which genes are present in the bacterial community and what they're doing. Okay, so this gives you a functional perspective of the microbial community. You can also extract the RNA from the cells, reverse transcribe it into DNA, and run it through a sequencing machine. And that tells you about which of these genes are being expressed, okay, which is important because genes that are not expressed, they're kind of silent, right? They're not doing anything. And that's called transcriptomics, okay? So this tells you about which genes are expressed by the bacterial community. And you can even extract other types of molecules like proteins or small molecules like metabolites and run them through a mass spectrometer, and this is what we call proteomics and metabolomics. And this tells you about what the bacterial community is making, okay, which metabolites are being produced. And so this collection of experiments gives you a multi-view perspective on the microbiome. So originally, when we only had 16S ribosomal RNA sequencing experiments, the data looked like this. So you had a matrix with your samples, where samples are different columns, and the different taxa or species are the rows, and an entry in the matrix is just the abundance of a taxon in a given sample. And now we have, this is a more modern view where you are not only characterizing the abundance of different taxa in each sample, but you're also characterizing which genes are present or which genes are being expressed through transcriptomics, as well as which metabolites you have present. A key feature of all of these data is that the columns in this matrix, so if you look at the column for different taxa in a single sample, or for different genes in a single sample, these are all probability distributions, right? We're interested in the abundance of a taxon in a sample. That's the sum of these entries would be 1, and the same thing for the sum of, say, expression levels if you want to be able to compare different samples, you have to normalize the columns so that they're in an equal scale. And the way you do that is you divide by the sum, okay? So we say that the data are compositional in nature. Each column is a probability distribution, so that's a peculiarity of microbiome data. But where do these distributions come from? Okay, how do we obtain this data matrix? So we apply a number of different bioinformatic pipelines. The raw output of a sequencing experiment is just a collection of short reads of DNA. Okay, these are shorter than what you actually get. What you get is typically at least 50 base pairs. And if this is a 16S ribosomal RNA experiment, these are all sequences that come from the same gene, okay, from the 16S gene. And so what you can do is you can cluster these sequences into what we call operational taxonomic units. Okay, so if you have a collection of sequences that all look very similar to each other, you can assume that they're all from the same bacterial species, or the same taxon. But sometimes those species will be known, other times they won't be known, and so we don't call them species, we call them OTUs. Of course, there's some intricacy in this step, so you have to understand how different two sequences have to be before you say that they're different species. If they only differ in a single base pair, does that mean that you have two different species, or could that be due to an error in the sequencing experiment? Because sequencing machines are not perfect. So there's some algorithm that's involved in doing this clustering, and then you can simply count the number of species that you have of each type and normalize into a probability distribution. This normalization itself is not straightforward, so you have different options. You can filter out rare species, you can normalize each count by the length of the read, okay? So there are choices to be made in this step as well. If you have metagenomic reads, you can do a very similar thing. Now, these are sequences that could come from any gene in the bacterial genome. And if you have a library of reference genomes, okay, so genomes for bacteria that have been characterized previously, these are bacteria that can be cultured in the lab, you can simply try to align each of these reads to one of the genomes in your reference library. So maybe this one comes from P. brevis, this one comes from E. coli, and that way you classify reads into different operational taxonomic units, and you obtain a probability distribution. You can do so this provides a taxonomic view of the bacterial community. Okay, so we're taking metagenomic samples and translating them into species. You can do the same thing with transcriptomic data. Okay, you can classify reads by species and then obtain a distribution over species, and that would be interpreted as the contribution of each taxon to overall gene expression in the community. Okay, so this is how we obtain these columns. You can do the same thing for columns that characterize different genes. Okay, so if you have a metagenomic data set, you can classify each of these sequences using some sort of gene ontology. So instead of classifying them by which species they belong to, we classify them by the function of the gene. And then, again, we can normalize this into a probability distribution. And we can do this with transcriptomic data as well as from metabolomic data. From metagenomic data. Yeah, but what about unculturable bacteria? So there are many bacteria that we can't characterize in the lab. You can't grow them in a lab, and you can't sequence their genomes. But they're still present in the samples that we have. So this is a bit like the dark matter of the microbiome. So you have, say that you have 3 different metagenomic data sets from 3 different samples. What I'm saying is that some of these sequences you won't be able to map them to a genome that's in a reference library. Okay, they've never been characterized before. So what can we do about these species? This is a very important problem, especially when you're looking at a very fine characterization of bacteria. So if you're trying to distinguish, say, different strains of the same species, okay, you need to resort to more complicated methods. And so what you can do is you can take all of these sequences and do some sort of partial assembly. Okay, so you align them relative to each other you can't do this over the entire genome, okay, because there's not enough coverage, but you can obtain collections of sequences that align with each other. And then what you can do is scan through the genome and identify positions that are highly variable. Okay, so this is an example of a position that is not variable, because in this position all of the reads have the same base, right? They're all a T. But in this position you have 60% T and 40% G. And so we call this a highly variable site. And so if you scan through all the positions in the genome and identify variable sites, you can tabulate the percentage of the rare variant in each sample and for each site, for each variable site. And then often there are patterns in this table that allow you to recover what the OTUs in the mixtures are, okay? Which species are present in these samples. So for example, if you see that these 2 sites always seem to have a 1-to-1 relationship, that would be suggestive that there is some bacteria, there's a single bacteria that has the rare mutation in this site and in this site. Okay, and so we can, using different algorithms, which I won't describe for the moment, you can deconvolve this into a sequence of OTUs which have where each column would be the mutation pattern for that bacterial species. Okay, and so each column in this table would be a mixture of the columns on the table in the right. Okay, so this is just another way that you can obtain distributions of species. Okay, so there's a complex sequence of bioinformatic algorithms that are used to generate this table, and I just wanted to give you a brief overview of them, so you know that usually when we start with this table, we assume that information is unquestionable, but actually there are many assumptions that already go into defining our data matrix. Okay, but this matrix also has more structure, right? So the samples in an experiment will be structured. So usually you have samples for different subjects, say 4 different subjects. And usually the subjects are partitioned in some way into a disease group and a control group. Okay, so most studies in microbiome research are observational, and you're doing some sort of cross-sectional comparison. So you have two groups of patients, say patients with irritable bowel disease and patients who are healthy, and you are trying to identify, for example, taxa that are differentially abundant in this group versus this group. Okay? A problem with cross-sectional studies is that they cannot establish causation, and also there's a lot of inter-subject variability. And so usually studies are designed in a longitudinal way. So for each subject we collect more than one time point. And the way the longitudinal studies are designed is very important. Okay, so usually what is done is, if you're interested in studying a disease like diabetes, you'll take a group of patients that are at high risk of developing the disease, and then you'll have patients that eventually develop the disease and patients that remain healthy. And you're trying to identify differences before and as well as doing comparisons between this group and this group. You're interested in identifying differences before and after the development of symptoms in patients that have the disease. Okay, so that's a typical design of a longitudinal study. Sometimes it's possible to do randomized experiments. That's rare. So it's possible that the yellow group is a treatment group that is randomized in some fashion. This is more common in animal models, of course, but it has been done in human studies to study the effect of antibiotics, for example, or different interventions that are not too drastic, like diets or hygiene. Okay, so that deals with the structure in the samples. But the variables, okay, the rows of the matrix also have some important structure. So first, the taxa, the bacterial species, are related through a phylogeny. Okay, there are taxa that are very close phylogenetically, they're close relatives of each other, and then there are pairs of taxa that are very different from each other. And depending at what level of this phylogenetic tree you're looking at, it can be very important to take into account the phylogeny. So suppose that the taxa that you're interested in are strains. Okay, so you're looking at a very fine phylogenetic classification. Then different samples, different columns in the matrix will tend to have a very small overlap of species. Okay, very few species that are represented in one sample will be represented in any other given sample. Okay. However, if you look at clades, okay, or families of related species, there will tend to be at least one representative of each clade in each sample. Okay, so it's important to understand to utilize this information about how different bacteria are related to each other. And then, if you have information about genes or metabolites, these are also related to each other through functional families. And they're related to the taxa through the metagenomic and transcriptomic data. Okay, so this is a very structured, very complex matrix. It's not just it's usually not enough to just do a principal components analysis of it. Okay. So some important questions that you need to ask when you're analyzing data like this. So what are the effect of all these pre-processing techniques? Okay, so how clean is the output of the bioinformatic pipelines that I briefly described? What is the best way to normalize the columns of the matrix? Okay, does that have an effect on the outcome of the analysis? And all of that is still unclear. Okay, so the analysis of this type of data set is in its infancy. Well, that's probably unfair. I mean, many people have thought about it seriously, but I think there are many questions to be resolved. Another important question is how to use prior information about the variables. Okay, so I said that variables are related by phylogenies or gene ontologies, metabolic pathways, and how do we incorporate that into an analysis? Another interesting question, which is more of a scientific question, is what is a better model for a disease? Okay, should we be trying to identify pathogens, so specific taxa that may be associated to a disease state, or is the disease more due to an imbalance of the whole community? Okay, or some sort of impairment of the metabolic function of the community. Again, in some cases, one theory may be more appropriate, and in other cases, another theory may be more appropriate, and these hypotheses require different models, different analysis methods. And then finally, how do we deal with this fact that the data are compositional? So when you normalize a column, that induces a negative correlation in the abundance profile for different bacteria. So if you think of an extreme example where you have 2 bacteria in the community, and you downregulate one of them, so you apply an intervention that will kill off one of the bacteria, that will increase the abundance of the second bacteria as well, right? So these are negatively correlated. And so but ideally we would want to, if you're looking for an effect, you want to identify only the bacteria that is suffering a change or that is causing an effect in a disease. You're not interested in these negative correlations could lead to some false discoveries. And so the interpretation of parameters becomes a little tricky when you have compositional data. Okay, so what kind of methods are used in practice? So by far the most popular approach involves using linear dimensionality reduction methods or ordination methods. And these come from the ecology community. And what they do is they simply take your matrix of taxa versus samples, for example, and translate that into a plot where you can see the samples in low dimensions. Again, this immediately lets you see whether there are clusters of samples, whether there are any sort of gradients that you can distinguish that may be associated to different observables. So this is a very useful approach, and there are techniques that have been developed in the ecology community to deal with different problems that I've already discussed, like how to use the phylogeny. Okay, so if you do multidimensional scaling and you use ecological distances that employ the phylogeny, okay, that take into account the phylogeny, this will give you a more meaningful result than doing, say, a principal components analysis. And there are also approaches to study this type of multi-view data where you have data about taxa, genes, and metabolites, okay, like canonical correspondence analysis, canonical correlations analysis. There's this whole battery of methods that have been applied. And all of these methods, what they have in common is that they're based on some sort of factor model, right? So you're taking your matrix and you're factorizing it into some low-rank matrix and plus some noise, okay? Each of these factors, the columns on the left or the rows on the right, represents a latent variable which explains some variation in the data. So everyone will be familiar with principal components analysis. And principal components analysis is, you can interpret it as a maximum likelihood estimator in the case where this error matrix is a matrix of independent normal entries. A generalized principal components analysis uses a different distribution for the error, which allows the error to have some correlation due to some correlation in elements in different rows and different columns. And the assumption of generalized principal components analysis is this error is a matrix normal distribution. It's the same as saying that if you vectorize this matrix, you have a normal distribution, and the covariance is this Kronecker product of two covariances. So the parameters u and v are a covariance for the rows of the matrix and the columns of the matrix. So u would encode prior information about the variables, so for example the phylogeny in the taxa, while v encodes the covariance of the columns. So you could have information about whether columns are neighboring in time, for example. Something else that you could do with factor models is add structure to the factors. And when you have structured factors, that leads to useful interpretations. So consider, for example, enforcing that the factors on the left are binary vectors that are sparse. Okay, so one way to interpret this is the variables that have ones, they define a cluster of species or genes that somehow have a common pattern of variation across the samples. Okay, so this makes the interpretation of the group of ones relatively natural. If you have a factor which is block sparse, so all of this is 0, and then you have non-zero entries in just one of the blocks, that would be a latent variable that is associated exclusively to taxa. Okay? Whereas if you have blocks that involve more than taxa, maybe taxa and genes, that could represent interactions between different types of variables. If you have a factor on the right which is binary, so that everyone in the treatment group has a 0 and everyone in the control group has a 1, that is just a fixed effect, right? That captures, this vector would capture the difference in all the variables between control and disease, for example. So, structure factors can give you more interpretable models. One of the problems that I described, which is this problem of de novo reconstruction for species that are unculturable, is also a matrix vectorization problem. So you have samples as the columns and variable sites on the genome as the rows, and you're trying to factorize this matrix into the outer product of two matrices that look like this, and the columns of the matrix on the left and the rows of the matrix on the right are associated to different taxa, different OTUs. Okay, so you can interpret a column on the left as a mutation pattern of a taxon that's present in the samples, okay, in these samples. And this column is the distribution of taxa in a given sample. Okay, so a column on this matrix will be a mixture, according to this probability distribution, of the different columns on the left. Okay, so this is the de novo reconstruction problem. It's just a specific kind of matrix vectorization problem, but we have a very specific structure. Okay, so we need the left factors to be vectors, and then we need the matrix on the right to be a column stochastic matrix. Okay, so in the last, I have just a couple of minutes, so I'm going to go fairly quickly. So I'm going to argue that Bayesian inference is appropriate for this type of model. So in some cases we have a reliable prior distribution and an error model. So think about the de novo reconstruction problem. The prior is fairly believable. Okay, it's not a big leap of faith. You can put a prior distribution on the parameters of the model. But even when the prior is a bit subjective, say for analyzing a regular data matrix, estimators that have good average case performance may be preferable to alternatives that are more robust, okay, when the assumptions are fair, especially when more robust alternatives are more difficult to interpret, or they may be a bit pessimistic. A Bayesian approach makes it straightforward to include prior information, and this is probably the most important reason to use it. And it allows you to tune the degree of influence that the prior has on the analysis. And finally, uncertainty summaries are interpretable if they're available. Okay, so here I reference some of my work, which involves a Bayesian non-parametric model for OTU tables, and it has the structure of a factor model. Okay, what makes it non-parametric? Well, we're learning the dimensionality of the factors as well as a number of taxa from the data. And it makes it possible to include some fixed effects. And so I'll just show you briefly some results that you can get from this type of analysis. So this is a study of the vaginal microbiome in pregnancy. Okay, so these are, each dot in this plot are samples, and the x-axis is time. And what you see is that there are certain women that have preterm births. Okay, they give birth earlier than expected, and the color indicates a different type of microbiome for different women. So this study posited that the microbiome could be a predictor for early birth. And so what we did is we applied our Bayesian model, and this allows us to put uncertainty regions in traditional ecological ordinations. So here, each of these regions is a credible region for one of the samples. And you see that some samples are actually quite uncertain because they have a low coverage. Okay, the number of samples is relatively low. And when you think about the uncertainty of clustering microbiomes into different types, you realize that when you do things in a Bayesian way, you see that one of the clusters really cannot be defined as such. Okay, it's really a collection of samples that are between different clusters, at least according to an ecological distance. Another application that we use this analysis for is the study of type 1 diabetes in children. And here we are using different fixed effects. We're interested in whether the age of the child has an effect on the microbiome, the nationality of the child. So this was done on children from Russia, Finland, and Estonia. And also the disease state. So there are some children which are healthy, and some children that develop molecular signs of the disease, and some children who are diagnosed with the disease. And so using our analysis, we can get patterns that show the influence of age, say, on the relative abundance of different bacterial clades with some uncertainty on them as a function of, in subgroups of the patients, like the Estonians, the Finnish, and the Russian patients. Okay, so in this particular study, we found that, well, there's a relatively strong effect that shows that bifidobacterium tends to decrease in age in children from Russia, whereas relative to children from Finland, whereas Estonian and Finnish children have a similar progression in their microbiome. Okay, this is another example of an ordination plot. Okay, so this is again a principal coordinates analysis using an ecological distance. And here I'm showing the projection of the species, and you can see, we can plot some sort of uncertainty region around each species. And the parameters in our model also make certain phylogenetic relationships between the species more apparent. So this is an analysis that's based on, in this lower triangle, we have correlations that are derived just by taking Spearman's linear correlation coefficient between columns of the abundance matrix, rows of the abundance matrix. And there's a very small correlation between them. This is due to the fact that the data are compositional. But if you use the parameters of the model, either for the fixed effect or for the random effect, there are strong correlations between bacteria that are similar phylogenetically. Okay, so this is just an example of one way of using factor models, okay? And having some information about the factors or the dimensionality of the factors. But the main difficulty with using a fully Bayesian approach is the statistical scalability. Okay, so Bayesian methods are simply not scalable. So we can apply this to a case where we have, say, hundreds of bacteria, just a few hundred different bacterial species, and again on the order of hundreds of samples. But if you're looking at, say, thousands of metabolites, or you're looking at bacterial strains, or thousands of genes, then this quickly becomes unwieldy. And the methodology that we're using, which is Markov chain Monte Carlo, it seems to go against the grain of an interactive data analysis in biology. A biologist never wants to spend, even if it's possible to do MCMC, over 3 hours, they just want to be able to click on something and obtain an ordination plot. So they're not going to wait a long time just to get these nice uncertainty regions. And so we need alternatives for Bayesian estimation in structured factor models. I think that's critical for the advancement of this of microbiome science. And there are different approaches that can be taken. Variational inference is something that some people have been working on. An alternative that seems promising is approximate message passing, which is a way of incorporating prior information about the factors in a factor model, which tends to lead to estimators which have lower error rates when the data are actually structured as you assume. Okay, so these are low base error rates. There are also very interesting horizons in microbiome science in general. So we just seem to be scratching the surface. So there are new experimental techniques that will reveal the role of other types of organisms that interact with the microbiome, the dynamics of interaction between host and microbe, different mechanisms of transmission and colonization that are involved in the development of the microbiome, and also the potential of personalized therapeutics. Okay, it's an area that's in its infancy, and we don't know how possible it is to develop therapeutics for the microbiome. Okay, thank you very much.
I guess we have time for just one question.
Yes.
Does it affect the factor model different level of species? So if you are doing analysis in the phyla level or family level or genus level, does it affect your model in the different species level?
Absolutely, yeah. So, I mean, if you're using the same error model, right? So if you have you're using a factor model and you're not changing the way the error works, then clustering the taxa at different phylogenetic levels will make a big difference. As I said, if you're using a very fine classification, there will be no overlap between different samples, and so you won't be able to say anything, right? And so if you're putting some sort of a model in the error that takes into account the phylogeny, then that could make clustering the taxa less important.
Then maybe we can move on to our second speaker, Pauline Wong from University of Wisconsin Medicine. And Pauline is going to talk about data